Hi everyone, I'm Tonisha Kearney-Ramos, and I'm going to pretty much piggyback off of what Travis was talking about, just building off of how we can use neuroimaging in develop, kind of, John Fedota's sort of area where it's like really digging into the mechanism part and how that can actually inform the clinical component. I have no conflicts of interest, no interesting financial things going on for me. So I'm just going to take a step back and recap a little bit of what he touched on and just give a really brief summary slide of kind of the two most common strategies that have been approached for substance use disorders in terms of neural approaches. And one is, so we know that cognitive control circuitry tends to be pretty hypoactive or suppressed. And so using an excitatory paradigm allows us to enhance cognitive control. And, the thinking is that it'll help people to have sort of control of their impulsive behavior. And then the other one is the inhibitory component, which is reward circuitry tends to be hyperactive, and it's talking a little too much, and it's very responsive to drug cues and kind of precipitates the seeking component of the drug of addiction. And so using a suppressive form of TMS, whether that's continuous theta burst stimulation or low frequency stimulation. And so I'm going to talk really about some of the work that I did in Colleen's lab when I was a postdoc in her lab, which was one of the first people to really start thinking about, let's just go directly to the reward circuit and use suppressive stimulation, with the ventromedial PFC being sort of the superficial target that we're targeting. And so the question was, nobody had really looked at that, so can we actually access the whole circuit? So for any of you that are familiar with the idea of with the figure 8 coil, there's, the depth focality sort of trade-off where you only can get so deep, but how do you actually access the whole circuit when there's subcortical components of that? And so we're really relying on the structural and functional connectivity. But since no one had really targeted ventromedial PFC, we had to actually demonstrate if we could do that. So the first question was target engagement. Can we actually elicit a response in the entire circuit when we're targeting the hub, which was ventromedial PFC? And so we used neuroimaging to demonstrate that. So we did a study, an interleave TMS fMRI study. So this is where you actually have an MRI-compatible TMS coil, and you're able to actually deliver pulses of TMS and then capture the downstream responses in the brain in a pretty synchronous manner. And so with single pulses to the ventromedial PFC, we were able to see where in the rest of the brain lit up. And in a sample of 51 cocaine users, so a pretty good-sized study, we were actually able to show that targeting the ventromedial PFC, we got robust bilateral responses in all of the downstream regions of interest in this reward circuitry. So we got the bilateral caudate and putamen, nucleus accumbens, as well as the salience regions that tend to co-activate with the reward circuitry, so in the anterior cingulate and insula. So we are able to actually get the whole circuit by targeting ventromedial PFC. But the next question was, okay, is there a component of the highway that determines how much of the signal actually propagates, right? So the communication, the information can only get down a highway that actually has the integrity for it to get down there. So this study was just using the same data, but actually we also had DTI data. So we looked at fractional anisotropy. So we were looking at the integrity of the white matter tracks along these different the downstream targets from the ventromedial PFC, and we wanted to know whether or not that was predictive of how much functional activation we got in the rest of the circuit, and we actually showed that it was related. So the less integrity, the less signal got down there. So that's actually really relevant for clinical populations because we know that alcohol, for instance, there tends to be a lot of white matter degradation, and people have been using alcohol for a while. And then there's other comorbid sort of conditions that can lead to white matter loss, and so that has implications not only in terms of how efficacious a therapeutic approach would be in terms of the downstream effects, but also whether or not there have to be adjustments made in the stimulation intensity or the duration of treatment to maximize the likelihood of actually getting the therapeutic response in the target of interest. And so the next thing was, okay, so we can access the circuit, but can we actually manipulate it in a predictable way, right? And so here we actually wanted to know whether or not we could attenuate the reward circuit. So that was the whole point, is we want to attenuate the hyperactivity in the reward circuit. And so we used a sandwich design where we actually still use the TMS, interleave TMS fMRI, where we're just pinging the brain and seeing what lights up. But we also had 2 different sessions for each subject, so it was a within subject design. where they underwent an active session of inhibitory TMS, so in this case, continuous theta burst stimulation, and a sham, and then we compared that across each individual. And so what we found, and then they went right back into the scanner immediately, and we pinged their brain again, and we saw whether or not we were able to suppress the response in the circuitry. And in fact, that's exactly what we found. So in a sample of cocaine users, we found that that same circuit, we were able to suppress activity after that, that single session of inhibitory stimulation. And we actually transdiagnostically demonstrated this because we also showed it in alcohol users as well. So this was promising. So we're actually able to manipulate the circuit in the direction that we were interested in. But obviously the main question is, does this actually have behavioral relevance? So does this translate to the behavioral engagement of the circuit? Right? And so we know that drug cue reactivity, which is the response of the reward circuit upon drug cue exposure, is actually predictive of relapse transdiagnostically. So for most substances of abuse, this has been demonstrated to predict treatment response. So whether or not somebody will remain abstinent or not, the magnitude of this response actually can predict the likelihood for relapse. So we wanted to know whether or not we were able to actually shift drug cue reactivity. And so again, it was a sandwich design, but in this case, instead of pinging the brain and seeing the immediate response, we actually did a drug cue reactivity task before and after the inhibitory session. And again, our hypothesis was that we were going to show decrease in drug cue reactivity in the circuit. And so in this case, we use functional connectivity because naturally, again, talking about transsynaptic modulation, we're interested in the connectivity from the target to the downstream regions. And so we showed in cocaine users that at baseline, they did show an elevated drug cue related functional connectivity in the circuit, and that this was robustly attenuated following active but not sham inhibitory stimulation. So this was promising, but we also actually showed the same thing in alcohol users. So there was a transdiagnostic reduction in the drug cue related functional engagement of the circuit. So this was also really promising. Now, one thing that I do want to mention is the fact that at the time when I was doing my postdoc with Colleen, we didn't see this same pattern with activation. So we saw it with the functional connectivity, so that was promising. But there was something going on with just looking at the activation. And so I want to mention the fact that there is this idea of there's individual variability in the level of response depending on how you're assessing that. Clinically, that's been demonstrated across a bunch of different psychiatric conditions, so eating disorders, depression, and so I'm already telling you the answer. Baseline brain state is predictive of your therapeutic response, and I'm going to show why. But it's critical that the reason why actually is dependent on the biomarker. So a lot of these other studies that showed variability in clinical response didn't actually measure the brain, the mechanism itself. So when they got a difference in clinical response, they didn't really have a reason why. So there's really an importance of actually, when you're developing therapeutic neuromodulation, to have a biomarker that you're measuring so that you can know and you can kind of gauge what's going on when you don't get clinical response. And I'm gonna explain why that's the case. So when we looked at, this study was where we looked at baseline and how predictive activation in the reward circuitry was predictive of how they responded to this inhibitory session. And we found that if you just look at the group at baseline, the active stimulation didn't look like it was doing much. The same was for sham, so there wasn't really a treatment effect when you just look at the group. But if you look at individual participants, we actually showed that there's variability in how responsive they were to the cues. So there's an element of cue reactivity. So people who were actually very responsive to the cues, they were the ones that showed the attenuation in the direction that we wanted, right? They showed the inhibition of drug cue reactivity. But people who weren't really that impressed by the cues or who had no cue reactivity, they actually went up. So if you remember that drug cue reactivity is predictive of relapse. So we're not trying to induce cue reactivity, right? So we wouldn't have known this. So when you look at the group, they cancel out, and it just looks like there's nothing happening. But actually there's a bimodal response, and it's actually clinically important. And we know that it wasn't regression to the mean because we didn't see that for the sham, right? So this was very specific to the active treatment. And if we actually look at how predictive baseline cue reactivity was to treatment response, we actually show that it was predicted 60% of the treatment response. So it wasn't a non-significant biomarker. This is actually very predictive of which direction somebody's going to respond in. So if we think about the clinical implications of that, it means that when people are looking at group results, sometimes they're seeing that there's no response, but really there is. It's just that there's subgroups of people that are responding, and actually the way in which they're responding is very clinically relevant. So this is just a very preliminary conclusions in terms of ventricular PFC. I think now there's some evidence, like the study that Hamed mentioned recently, where they're showing that frontal pole is an interesting site in general. But at the time, Colleen was really the first person to start looking at that. And so, we were like really making case for like, yeah, this could be really an interesting approach using inhibitory stimulation for frontal pole. I don't think I have to convince you of that as much now, but that is something that we should consider looking at since there's so much research in DLPFC. And then also including neuroimaging is very, very important for therapeutic neuromodulation. It's a brain-targeted approach, so you can imagine how important actually incorporating mechanism in determining the optimal sort of clinical approach is going to be. And so I'm also making a plug for including neuroimaging and how important that really will be for moving forward. And so I do want to just acknowledge Colleen's lab, which is where I did my postdoc, and then my current group over at Columbia. I work with Meg Haney, she's my primary mentor, and then also Diana Martinez, who does some neuromodulation research as well. So yeah, that's all I got.